Now, scientists from Japan, in collaboration with 3 pharmaceutical companies, have developed a radioligand that facilitates the imaging of alpha-synuclein aggregates in patients with MSA. Their findings have the potential to completely change the scenario of diagnosing neurodegenerative diseases. Alpha-synuclein is a neuronal protein involved in functions like vesicle trafficking and neurotransmitter release. It is typically found in abundance in a healthy brain. However, the aggregation of alpha-synuclein has been closely linked to several neurodegenerative disorders, including Parkinson's disease, multiple system atrophy, or MSA, and Lewy body dementia. MSA is a movement disorder that also affects the autonomic nervous system, which controls essential functions such as movement, breathing, and digestion. Thus, the imaging of alpha-synuclein aggregates in vivo, or directly in a living organism, could be a potential diagnostic confirmation of MSA. However, the road to the live imaging of alpha-synuclein has been marred with obstacles, including the lack of sensitive imaging agents. Now, a collaborative effort by researchers from the National Institutes for Quantum Science and Technology, including Dr. Makoto Higuchi and Dr. Kiwamu Matsuoka from the Quantum Life and Medical Science Directorate, Institute for Quantum Medical Science, has completely changed the scenario with 3. The team's findings have been published in the journal Movement Disorders. Prior to the clinical assessments, in vitro studies on the binding properties of 18-FSPALT06 had been conducted on the postmortem brain tissue of patients with MSA and healthy individuals, showing promising results. For the first-in-human imaging studies, the researchers enrolled 3 patients who were clinically diagnosed with MSA and 1 72-year-old healthy control, HC. Among the 3 patients with MSA, 2 were identified as having MSA with predominant Parkinsonism, MSAP, and 1 with MSA with predominant cerebellar ataxia, MSAC. PET scans with 18-FSPALT06 was performed on all the patients and specific binding was estimated by the radioligand retention in the tissue. Remarkably, we observed enhanced 18-FSPALT06 retention in the putamen, pons, and cerebellar white matter and peduncles of the patients with MSAP and MSAC, in sharp contrast to minimal radiosignals in the corresponding areas in the brain of the HC, explains Dr. Higuchi. The researchers also found that 18-FSPALT06 has a high affinity for MSA-type alpha-synuclein aggregates and that it does not cross-react with other off-target components, indicating its high specificity and consequent potential use as a probe for MSA diagnosis. With respect to the long-term applications of their work, Dr. Higuchi and Dr. Matsuoka share, "We are encouraged by our findings, and investigations into the visualization of alpha-synuclein aggregates in other alpha-synucleinopathies are currently underway." Kiwamu Matsuoka et al. The National Institutes for Quantum Science and Technology. 2022, August 30. Researchers visualize alpha-synuclein pathology in living patients with a neurodegenerative disorder. https://ift.tt/GISCM8F. Accessed August 30, 2022. Thanks for watching. Please subscribe my channel for more videos.